Welcome everyone. So this is looking at COVID and what biomarkers we have actually tested or others have tested in cases of COVID and what we understand about the pathophysiology of the disease. Just to say, there hasn't been any publications or any data which has been shared about the CSF findings in COVID. And I think it's secondary to this myth which is circulating that it doesn't involve the brain. It's now becoming apparent as more data filters out of China and also out of Italy that this is not the case. So this image from the ACS Chemical Neuroscience Journal in fact typifies what happens in COVID. So COVID is named COVID-19 after the fact that it came through in the year 2019. In fact, it's very similar to the SARS COVID virus, which is the original COVID virus which caused an epidemic. And as you can see, it has these spike proteins, and predominantly everyone develops a respiratory infection. It initially goes in the nasopharyngeal region and tracks further down. But what's apparent is that like SARS, it may in fact spread directly via the nasal passages into the brain, or also through retroneuronal spread, or through hematogenous spread. So these are working hypotheses, but no one's in fact confirmed it. With SARS, there were publications of autopsy cases showing that there is CNS involvement. So what do we have? And the data is very limited, unfortunately. So if we look at the hypotheses, it's uncertain how much of the central nervous system is affected at the outset. Often with flu illnesses, influenza in particular, and also coronavirus infections, people become quite dizzy and also develop some level of confusion with it. But in terms of mechanisms, the hypotheses are that the injury is probably from direct viral infection, or it may be secondary to all the systemic effects which arise from the infection, and these could be either metabolic or hypoxic damage to the brain. More recently, some of the deaths being attributed to respiratory failure were thought to be, in fact, due to brainstem involvement by the virus, so the so-called Ondine's curse case, which many of you may have come across on Twitter. This is all preprint publications, by the way, so they're pre-review or peer review, and but they are interesting in helping us understand about what we understand insofar as the neurological presentations are concerned. So this is a publication from Wuhan itself, which has been the epicenter of the infection. And what you can see is that you have about 214 cases from the 3 designated hospitals for COVID admission from Wuhan. And in that, 36.4% had some form of neurological involvement. Now, you might have read in the JAMA publication this morning that hypertension seems to be a major risk factor. And insofar as the cases which they've classified, central nervous system involvement was predominantly through cerebrovascular accidents. So you're looking at infarcts as well as hemorrhages. There was also peripheral nerve involvement, and they do interestingly include anosmia or hypoanosmia in this. So the virus obviously is spreading through the nasopharyngeal spaces. There's also some skeletal involvement, so rises in CK has also been noted. So if you look at the peripheral blood analysis, which has been published in patients with CNS involvement, what's been characteristic, and not only from the Chinese publications, is that there's significant lymphopenia in these individuals. And if you look at the drop, this is total lymphocyte counts, by the way. And I'll go through on the next page another publication which looks at relative lymphocyte counts and subpopulations as well. So lymphopenia seems to be quite an issue in patients with COVID and the CNS involvement. And also there's a drop in platelet count, which may be from as a secondary result of multi-organ failure, which can happen in these cases, as well as a rise in urea, where you get a lot of infections involving the liver and the turnover of products. So what's marked is that in those with severe disease category, these were found to be more significant in their drops. Now, going from a different publications published by a neighboring city, which looked at about 103 cases of COVID-19 infected patients, and they compared it to a control group of 22 non-COVID-19 pneumonias. And what I'd like you to draw your attention to is the subpopulations in the lymphocyte. And this is quite interesting. It is behaving in a way which you would expect from a virus which is directly interacting with the lymphocyte subpopulation. So viruses which do that are those which sabotage the immune system by overwhelming it. And you can see in the larger population group, this is the case as well. So in the non-severe, which was 71 patients, you find that the CD4 T cell total count drops much worse in the severe compared to that in the mild and asymptomatic individuals. And then in also the similar pattern is seen with the CD8. Now the CD19, which are the B cells, are also affected. So the adaptive immune system seems to be compromised in patients infected with COVID. Now, in terms of when you think about where immunity arises, this is one of the first defenses which you have against viruses and being able to recruit the rest of the immune system into the fight. So we hear about the cytokine storm being an issue and the cause of the lung problem, but this data actually questions it, because to actually have the cytokine storm to be effective, you need a sufficient lymphocyte population, and also you need sufficient NK cell response as well as neutrophil and monocyte. And interestingly, that latter population, the CD56 NK cells and neutrophil count and monocytes, are unremarkable. So that's a very strange presentation in these patients. And it raises a question whether we are dealing with overactivity of the immune system as being touted to date, or in fact, an immune system which is compromised by the virus. And you get a further impression that, in fact, what you're dealing with is direct viral damage. If you look at the indices of organ damage against the CD4 T cell populations, and what you see is that indicators like liver dysfunction, which is LDH and AST, drop as the immune system recovers, or that there is better immune system to be there. And sodium hyponatremia is better when the immune system cell count is better, and so is the hypokalemia. So this alludes to that that organ damage is in fact negatively correlated to immune system CD4 T cells. So the virus is probably directly attacking the immune system. And if you find that there isn't a proper cellular and humoral immunity, then you're more likely to get a compromised secondary response as well. Now, A lot of people have been talking about parallels to the original SARS coronavirus. So SARS being a severe respiratory distress syndrome, which it causes. And it's been found that the receptor, which is infected by the COVID-19 virus, as well as the original SARS-CoV virus, is the same. So they're both attached to the angiotensin converting enzyme type 2, which is present in multiple organs. So we know that it's present in cardiac, we know that it's present in kidneys, and we know it's also present in the brain. And this workup has been done extensively as far as SARS coronavirus is concerned. And they've shown, I've taken a slice of what's presented in the previous papers on SARS coronavirus, which shows the distribution of viruses in the brainstem of naive mice versus those who have received the infection. And these mice have had the human angiotensin converting enzyme receptor inserted by transgenic changes. So what we understand from the SARS virus is that it enters via the olfactory bulb, and there's rapid transneural spread to areas in the other areas in the brain, and that usually results from dysfunction or death of the infected neurons. And also some of these are located in the cardiorespiratory centers in the medulla. So if we transfer across what we already know, then we should really be looking at the brain in patients with respiratory dysfunction. To date, we've always thought this was a pulmonary failure rather than something happening in the brain as well. So despite my best endeavors asking multiple people what they know, we haven't as yet any CSF evidence or any autopsy of brain slices to show if there is COVID-19 in brains of patients who were severe and therefore died as a result. There are studies underway, and one of them is in Italy to look at neurofilament levels in patients infected with COVID-19 and to see if this could be used as an indicator of prognosis. So one of the ways to look at this in greater detail is to look at all the biomarkers which have been well studied and been linked to inflammatory mediated neuronal damage. And these biomarkers are well validated and well studied. And the number one top would be to look at neurofilaments. These are neuronal structural proteins. So when you have neuronal injury, they can be used to risk stratify how a person would do. The other is look at the innate immune system, which is there in the brain, how it responds. So if the GFAP activity increases, then it would be a prognostic marker. Osteopontin, which is also an inflammatory marker, should be looked at, and others which look at the innate and adaptive type 2 immune responses, the chitinase and chitinase-like proteins. Soluble TREM2 has also become an important thing, particularly in Alzheimer's disease as a marker of microglial activity, but it's another way of looking at what's going on in the brain. So these are studies which do need to be done. Unfortunately, they're probably not going to be available at every laboratory, but more on the research end to understand what the prognosis of patients are. So if there is an overwhelming inflammatory response in the CSF of patients infected by COVID-19, then it makes sense to obviate that and give steroids, for instance. Sometimes looking at the peripheral compartment is not enough to judge whether what's happening in the CNS is the same or it's separate. So moving on. This is just a quick idea of how we have been using it in MS. To your right is an MS patient in their 30s with active disease and early signs of brain atrophy, as you can see. This is a coronal section of a T1 brain scan, which is showing very high levels of volume loss and inconsistent with the age. And you can see that the neurofilament light chain level in the CSF is 7,194 picograms per mil. For his age group, it would be definitely less than 300, being the normal turnover of neurons with aging. Obviously, everyone can't have serial lumbar punctures, and this is not something which is advised, and we should be able to substitute in blood neurofilaments when we can. This gives you an idea of how blood neurofilaments can be used in profiling, for example, in an MS patient. So you've got serum NFL in the y-axis and weeks over treatment on the x-axis. And what you see is a rise and a peak at about week 48, which then comes down at 96 weeks. And you notice that there is an increase in gadolinium enhancing lesions, as well as a drop in SDMT, which is a marker of cognitive function, which then subsequently improves. And this is a real-time observation of what is a cognitive relapse. Now, neurofilament is a bulk biomarker, it's not disease specific. So ALS is one of the conditions where it's elevated at very supranormal levels. And this kind of just tells you what you may expect when there is massive neuronal damage. So you have here on your left a good correlation between serum NFL and CSF NFL. But if you look at the progression rate between two centers of samples, the progression rate increases as the blood NFL goes up on your x-axis. So these are probably one of the first biomarkers which we should be looking at in patients infected with COVID who may develop an encephalitis, but also a good way of monitoring our patients if they are becoming unwell with the concurrent viral infection. So one of the logistic difficulties which we've faced in our department since the epidemic starting up in London as well is how to do these kind of investigations. We should be really prioritizing clinical need and also keeping our patients away from hubs, which would be a source of infection spread, and hospitals are classic for this. And as we face more and more difficult problems over the ensuing months, we should think about how to do the investigations which we do need to do in patients. So as you all know, we do provide CSF neurofilament testing, and these are some of the logistic difficulties which we've had to contend with since setting up the test. And one of the things we were thinking about is using telemedicine to see if what test is needed. So case selection by a physician picking to say, okay, this is the presentation of the patient, this is my question. And then the procedures are done locally. So you don't, in fact, send people in to big centers to have their LP or their blood test, but you do it in an outpatient setting. So this is at a distance away from where patients would be at higher risk. This may be an opportunity to think about using lumbar puncture nurses, which are used elsewhere in Europe. Then the samples can be couriered during the same day, usually within a distance of 300 miles. And then you have a central hub analysis of the markers of interest. This reason for this is because the turnaround times are improved, but also the reliability of the results improves. What we're seeing at the moment is that a lot of laboratories are trying to set up the PCR test for COVID, producing a lot of variable outcomes, and the sensitivity of the PCR from the nasopharyngeal swab stands up average on 72%, whereas the sensitivity of just doing a CT chest to see if a person is infected is 98%. So that's room for improvement. And when you do these kind of investigations, you need to make sure that you have less variability. Now, once that's happened, it's also a good idea to who is of interest or who is interested about the actual information which has been gathered. So at the moment, we're living in a black hole as far as what happens to the brain with COVID, but it may be that over the next few weeks, the data will start filtering in where we can have a better assessment of it. So in summary, I think it's quite clear that we don't know what's happening in terms of CNS involvement in COVID-19 cases. The peripheral blood analysis definitely shows that there is lymphopenia, which may be critical and may in fact point to more severe outcome. And this also suggests that there is a direct role for viral injury, which may be relevant to the disease. Now, we need to look at how to start doing the work to figure out if we can risk stratify how patients with COVID infection do, but also need to think about very inventive ways of carrying on with our existing work, which seems to have ground to a halt as a result of the pandemic. Okay, I will close there and take any questions.
Sharmilee?
Yeah.
It's Gavin here. How are you?
Hi, I'm well, Gavin. How are you?
Yeah, so one of the things that worries me is that the ABN and the Italian guidelines have kind of just said natalizumab safe. So I'm just worried from a theoretical perspective that it may not be safe if you've got this penetration of this virus.
So Gavin, I mean, the issues about natalizumab is the idea that it's safe because it inhibits just transition of immune cells into the CNS space. Obviously, in a situation where there is lymphopenia developing because of the infection, I do think you may inadvertently increase the risk of the underlying PML risk. So that's a major consideration as far as natalizumab is concerned. I suppose if you did extended dosing, i.e. instead of the 4 weeks, you do the 6 weeks, this would be less of an issue. But this does need to be considered. Obviously.
So what I'm actually worried about is that if you do get, say, a neurotropic strain or the virus gets into the brain and causes an encephalitis, because you're not immune surveilling the nervous system, you may get a very strange encephalitis, like a slow grumbling encephalitis.
Yes, so that's a possibility. The virus in itself may create a response in there, so that will lead to a poor outcome. The other potential is that the microglial response, which may still be evident in the brain, may be over-enthusiastic. So you may get real massive inflammatory responses or gliotic responses. So difficult to say. I think this is going to be very much case dependent in terms of figuring out what's going to happen with natalizumab treated patients if they do acquire COVID at a sufficient load that they become severely infected.
So this is one of the reasons why we're going to have to promote that register study, so we get everybody into that study so we could answer these types of questions. Yeah.
Thank you.
Any further comments or questions from anybody? If not, there will be the facility to be able to ask questions at a later date on the one that is uploaded on the website. So thank you very much, Sharmilee.
Yes, no problem.
I'll come back to you with any future comments that we'll have from others that will be able to watch it once it's live on the website, which should be hopefully later on today, or first thing, which should be later today.
All right, thank you very much.
All right, thank you everybody.
Okay, bye-bye.
Cheers, bye.
Thank you, bye-bye.